Hello, my lovely anatomists and physiologists. Michele here. Welcome to another Chapter 14 Special Sense video, and we're focusing on the sensations of the inner ear. So we just finished talking about hearing, and now we're talking about both gravity and acceleration. Now, both of these are being collected at the vestibule, which is this region here of the inner ear. So remember, the inner ear has three parts. It has that snail shell looking cochlea. It has what might be described as like the body of the snail. That would be the vestibule. And then it has those three semicircular canals. And so we're looking at this kind of space in the middle of the inner ear called the vestibule. And remember, the vestibule is going to have both that bony labyrinth, the perilymph, the membranous labyrinth, and then the endolymph. So when we're looking at the actual organ that's detecting gravity and acceleration, it's inside that membranous labyrinth in that area called the vestibule. And so macula is the singular form of the word. Maculae is going to be plural, and these structures are located inside what's described as the utricle and the saccule. So over here in this particular piece of our model, we can see our cochlea. They make that a little bit more visible here. So we can see our cochlea here. And then actually cut is the semicircular canals. So they're actually broken off of this particular model. And so this area here is showing our vestibule. And so here you're seeing the saccule. And then here you would see the utricle. And this model up above, which is the same image that we've been looking at in previous videos, we can see the cochlea. We can see up above the semicircular canals, and then we can sort of see in this space here the vestibule. What we want to pay attention to is this particular diagram that we're getting out of our OpenStax anatomy textbook, where we have the maculae shown here. The maculae, AE ending as plural, macula would be singular, and we're really just looking at one. We're going to see again our hair cells like we had in the cochlea, but notice this time that you have not only those stereocilia coming up out of the hair cell, but you have this one super long one, and we call this super long one the kinocilium. And that sort of gives like a directionality to the cell. So if it's bending towards the kinocilium or away from the kinocilium, that's giving us some important information. You have those stereocilia and kinocilia of the hair cells embedded in that gelatinous material. And sitting on top of that, you have these otoliths. And the otoliths are going to be calcium carbonate crystals. So they're actually little solid molecules sitting on top of this gelatinous material, and we can call that whole structure the gelatinous material in the otoliths. We can talk about that as the otolithic membrane. And then notice your hair cells are your sensory receptor cells, so they're going to be synapsing onto that sensory neuron. And what we're seeing then is in that maculae, so above the maculae right here, you have your endolymph, right? And so the idea here is if you tilt your head forward, the endolymph fluid is going to move. That's going to pull with it the otolithic membrane. So that's that gelatinous material plus those calcium carbonate crystals, those otoliths. And that's going to cause a bend. And you can see that in the picture. That's going to cause a bend of the stereocilia and the kinocilium. And that is going to stimulate the mechanoreceptor of the hair cell. So let's take just a minute and go back up to our table that we've been filling in. When it gets to gravity and acceleration here, I would actually list the maculae as my structure. And then I think I would say vestibule, inner ear. You know what, I'd like to go and do maculae of the vestibule in the inner ear. And I would actually like to make a notation back to our hearing and actually say like spiral organ of the cochlea in the inner ear. And then when we're doing the gravity and acceleration receptor cell, I would like to call that my hair cell. So we call it the same, even though we have that difference in terms of the presence of kinocilia. To finish up our discussion here, we do want to go through our pathway. So when we're looking at our gravity and acceleration for our pathway, basically we can say that the head tilts, and when the head tilts, then we're going to have movement in the endolymph, and gravity will pull on the otolith. Otolith is talking about, again, the whole gelatinous material and the calcium carbonate structures there. And so, or maybe I should go ahead and say otolithic membrane. And when we are getting the otolithic membrane to move, then we get the bending of the stereocilia, kinocilia of the hair cells. And so this can either depolarize or hyperpolarize the cell. And remember that you're getting information on both the left side and the right side. So that information is coming together into the brain to be interpreted. And so this is going to send the signal to the sensory neuron, which is then going to be part of that vestibulocochlear nerve. This is going to go to the brain stem, and then we're going to see it go to two places. I kind of want to number it, but I really shouldn't do that. So we're going to the brain stem and then to the cerebellum. Remember, your cerebellum is like that part of your brain that smooths out your movements, that controls like your muscle memory type of movements. And so clearly you would need to have this information about the position of your head, about gravity acceleration as you're smoothing out those movements. And then we're going to get the conscious regulation. So from the brainstem, it goes to the thalamus. And then of course that will take the information to the cerebrum. That's it for gravity and acceleration. Stay tuned for rotation. And as always, take care of yourselves and each other.